In red, we have an origin of replication. All vectors should have an origin of replication. It is the sequence that has a very specific set of properties, usually a specific number of A's and T's in one region, specific number of G's and C's in another. But these specific properties allow it to de-anneal during replication, which allows DNA polymerases to get into it and to start the replication process. Because otherwise, there's no way for them to actually start. They can complete it very easily, but you need an origin to actually start. This will always be present in your vector, and unless you're designing vector plasmids, you don't need to worry about it. But there are a couple of things to note. For one thing, you cannot have multiple origins of replications that are the same in the same organism be stable. So I couldn't have two plasmids with one origin, with the same origin of replication. They both have to be unique. If they are the same, they compete for the same enzymes that aid in replication, and one of the plasmids ends up becoming dominant and the other one gets lost over time. So it's quite important that if you're doing double transformants, you use unique replication sites. There's also a number of different types of replication sites. So a common one is Col E1, which is the most commonly used origin of replication in E. coli. So that's often used to replicate bacterial cells. There's also the F1 origin of replication, and that's used for phages. So filamentous phages specifically will use an F1 origin of replication to replicate. And then there's a few others. So, for example, there's an SV40 origin, which is found in plasmids that are meant to reproduce in mammalian cells. And those are very important because mammalian cells normally don't harbor plasmids. But you don't have to worry too much about it. Just make sure that if you're trying to put it into a certain biological agent, it has an origin of replication that is suitable for that biological agent. There's no point trying to shuttle a plasmid through E. coli if it only has a phage origin of replication.